1919 was an auspicious year in history. The First World War ended, ushering a drastic change in the world order. The Philippine Islands had been a U.S. territory for 2 decades and was promised independence under the Jones Law of 1916. Progressive reformers saw the country and its people as a major target for modernization, and education was its main weapon. 7 forward-looking Filipinas came together in 1919 to create a school where young women could gain the knowledge and skills that would make them modern women. Paz Marquez Benitez was the first president of the Philippine Women's College. Jose Abad Santos was its first chairman of the Board of Trustees. Francisca Tirona Benitez was the second president, and with her husband Dean Conrado, guided the school from a house on A. Flores Street to its iconic Taft Avenue campus. In 1932, the college became a university, making PWU the first university for women in Asia founded by Asians. It provided a space where innovations in education flourished, and young people were encouraged to be the best that they could be. For over 100 years, the Philippine Women's University has been known as a leader in quality education. In 1934, PWU moved into its main campus on Taft Avenue, and since the 1970s has been co-educational. Located in the heart of Metro Manila, it is easily accessible by public transport and surrounded by affordable housing. Today's PWU offers undergraduate and graduate courses in several fields of study. Its business and management programs are responsive to the needs of industry, using evolving technology for global competence. PWU graduates excel in arts and sciences, education, social work, and diplomacy. Its fine arts and music programs have produced outstanding graduates through a holistic education that treasures heritage as well as excellence. PWU has pioneered in fields such as food science, nutrition and dietetics, medical technology, pharmacy, and nursing. PWU continues to play an essential role in producing graduates who possess the skills that make them competitive in the country and anywhere in the world.
We Patriots are one big family.
Being part of the PWU community strengthens us.
Through the years, we support each other through good times and bad times.
We Patriots always have each other's back.
It's a big honor to be part of a community that is open to the ideas of each other.
drives us to excel and be the best version of ourselves.
That's the standard of excellence that we Patriots strive for.
I'm thankful that this community lets us be who we want to be.
We thrive because we accept each other for who we are.
And like a family, we treat each other equally.
Actually, PWU gave us a home and helped us to be a patriot in society.
We may have different individual drums and instruments,
but we march to the same beat together as one united community.
Do you want to march with our community of patriots?
I am a patriot.
I am a patriot.
I am a patriot. I am a patriot.
The approach to science is research. Otherwise, there is no science at all. Welcome to the Philippine Women's University School of Medical Technology Research Forum 2023.
Happy Chinese New Year of the Rabbit. May this year bring peace and prosperity to us all. So now let us begin the year right with a proper mindset and how to find our way in the completion of a thesis research.
We would like to thank each one of you for taking the time to join us in this endeavor. Let us begin with a moment of silence and place ourselves in the presence of the Almighty for the invocation. This will be followed by the national anthem.
buhay at sa langit bumbaw, kay dilagang tulad, awis at paglayang minamahal, ang kisap ng watawat mo'y tagumpay na nagdidigdig, ang bituin at araw niya kailan pa may di magdidinig, lupa ng araw ng luwal, at ipagsitabay langit sa piling mo, aming ligaya na pag may mga api ang mamatay ng dahil sa'yo.
Ladies and gentlemen, here to welcome and to present the objectives of this webinar is the Program Chair of the PWU School of Medical Technology. A round of applause to Dr. Sherwin B. Torian.
Good afternoon, ladies and gentlemen. To our University President, Sir Marco Alfredo Benitez, Senior Vice President for Academic Affairs, Dr. Filina Siyang, the academic heads, administrative officers, the faculty and staff, representatives from various academic institutions, to all our students and researchers, good afternoon. Today's research forum is appropriately themed as Research Thesis Scientific Journey from Title Through Gaps. Our speakers shall explore the possibilities and offer invaluable insights on how to come up with a suitable medical laboratory science thesis title and the sources where you may find the inspiration to pursue a worthwhile research study. We have invited Dr. Rosana Tala to lead the way as she elaborates on the thesis title, Areas to Explore for a Medical Laboratory Science Research. A lot of students are having a difficult time figuring out and constructing research title that they could feasibly complete to fulfill the requirements of the program. Allow me further to share that knowledge or research gaps should not be a roadblock in finishing the thesis, but a gem of opportunity that when surpassed will offer a depth and fresher perspective to the research study. We have also invited thesis presenters from our college and master's programs to share their research theses that may impact our hopeful future researchers and guide those who are already immersed in their respective studies. In a broader sense, this event aims to, first, elevate the standards of medical technology research theses by enriching the awareness of students preparing for and already conducting their studies for a better, successful, and more contributive thesis in the field of medical laboratory science. And secondly, to strengthen collaboration among students, academic practitioners, and other professionals to build a positive and reinvigorating culture of research through the sharing of best practices and relevant experiences. I warmly welcome you all to the first research forum in medical laboratory science for 2023. I enjoin everyone to listen to the discussions and be enlightened. I am affirmative that after this webinar, those who are coming up with a thesis proposal may be motivated and more knowledgeable. Once again, welcome to the School of Medical Technology. Have a pleasant afternoon and best regards.
Thank you, Dr. Sherwin Toriano, for your warm welcoming remarks. This webinar is indeed a very promising, not only because of our guests and speakers, but because our virtual activity is graced by the presence of the Director of PWU Graduate School. So ladies and gentlemen, please welcome Dr. Emerita Zornosa.
Staying motivated can be difficult for researchers. The failed experiments, negative results, and lack of usable data can leave the researcher feeling discouraged. Perseverance. People say perseverance without motivation is often short-lived. Without motivation, Work can leave you feeling unsatisfied and uninspired. Please do not let this happen to you. Good morning, everyone. Let me welcome you all to our 2023 School of Medical Technology Research Forum. Together with our guest speaker, Dr. Rosana Tala and Dr. Sherwin Toriano, our Dean of School of Medical Technology, help us to welcome you all. Today, I was given a task to provide you with some tips on how to stay motivated to do your research. Let us start by joining our hands and keep ourselves a round of applause for bringing this research forum a success. Yours truly is Dr. Emeritus Sornosa, currently your Director of Graduate Studies at PWU. My first sharing are the 4 tips on how I kept myself stay motivated and keep working towards my goal to research success. First, remember why you do your research, why you are into this project in the first place. Perhaps you hope to make a difference through your research. Young scientists often start with the ambition to find a discovery that can change the world, unaware of how difficult the path to a breakthrough. Please remind yourself, why are you working on this research, and that everything you do and every step you take is important and serves a purpose. Second, take small steps and reward yourself. Research is a long journey to the result. Waiting for a reward is the ultimate goal, such as publication, can be very tiresome. Set a small milestone within your journey and celebrate reaching them. This will make your process itself motivating. If your goal is to finish writing your thesis, reward yourself for writing every 10 pages. If your goal to publish a paper on your project, celebrate its completed sets of data that you have gathered. Third, expect imperfection. The truth is, researchers encountered failed experiments, non-statistically significant results, or conflicting data more open than they would like to admit. This is the reality of research. Expecting that some things will not go perfectly as planned, prepare to adapt to the situation, prevent you from getting discouraged, and help you stay motivated. Fourth, work smart. Working smarter instead of harder will let you tackle your tasks more efficiently without adding more hours to your workday. The researchers who do this can stay motivated for longer in their careers. One way of working smarter is choosing efficient technologies and let you complete experimental procedures faster without compromising your results. Make the most of your Google platforms, Google Sheets for monitoring and tracking your work, Google Drive for proper storage, Google Form for your survey questionnaires, Google Docs as your editor, and so much more. You may even use Google Clouds. The available and efficient tools and technologies for life science research can help you accomplish more in less time and with less effort by making smarter choices for the tools you use in the lab, including cell isolation and culture technologies. My second sharing for today is the 4 easy habits needed to your research success. ACRA, ACRA. First, attention. Your research needs 100% attention that will allow to register into your mind the learning input you require. Have you recalled once your professor shouted inside the classroom, "Please pay 100% attention"? Have you realized that our professor is correct? What about the term input addiction? Do not allow this to happen, as you may be able to put junk in your mind. Because if you entertain shallow learning, there is a tendency of you forgetting everything instead of getting something. Second, comprehension. Comprehension is understanding the idea to get the stored idea or knowledge in your memory. Ensure to understand something into 100% comprehension, and this is called deep learning. First, you need to understand the thing before you can remember it. Learn to associate your learnings and always have your checkpoint. Do not forget to test yourself if 100% understanding is there. Never 99.5, never 99%. Please ensure 100% understanding. Try to develop a checkpoint every day. Develop a 30-minute you time. Maintain a daily journal reviewing what happens each day. Recall. Do not end your learning without the recall sessions. Have you heard of this? The teaching activity is the best way to learning something. In creating a group or team and sharing sessions on whatever learnings you gain for the day, for the week, or month can help you test yourself. And the last, application. Research can transform your life. An application of what you learn in your program can make you realize that research has a purpose. This is the application of all your learnings. We need to connect whatever learnings and resources gained and run to get that research done. Why do I say run? Develop a time calendar with energy. Your content organization is required to get the correct results or output on time. To end, let us summarize and recall if we gain the learnings on how to have the right motivation in your research success. The first 4 tips. Understand always the why. Take small steps and reward yourself. Expect imperfection. Use it as a challenge. Work smart by using efficient tools and technologies. The second tips. 4 easy habits to develop. First, attention to your learning, 100%. Switch off notifications, refrain from distractions, have the 100% focus on your learning. Second, comprehension. Understand to remember, learn the association of your learning, and always have a checkpoint. 3, recall. Teach and share. Be able to check if you have the recall. 4th, application. Always have the result in mind, and when you apply your learning, this can help transform a result that will transform you. Thank you. Wish you success in your research journey. The PWU community from the School of Medical Technologies is joining you to success to your research. Good day.
Thank you so much, Dr. Emerita Lornosa. To introduce our first plenary guest speaker is the Dean of the College of Medical Technology of San Juan de Dios Educational Foundation, Incorporation. She obtained her Master of Science in Medical Technology degree from the Philippine Women's University, Master of Arts in Teaching major in Science, and Doctor of Education major in Educational Management from Eulogio Amang Rodriguez Institute of Science and Technology. Our esteemed guest speaker is also the research coordinator, panelist, and advisor to student researchers at San Juan de Dios Educational Foundation, Incorporation. To elaborate on thesis title, Areas to Explore for a Medical Laboratory Science Research. Ladies and gentlemen, please welcome Dr. Rosana Tala.
Good afternoon, everybody, and to all present here. First of all, I would like to thank the PWU College of Medical Technology, headed by Dr. Sherwin, for this opportunity to share with you my topic on areas to explore for medical laboratory science research. At the same time, I would like to extend my gratitude to my former mentors, Dr. Nini Lim, my thesis advisor, Dr. Gab, Dr. Silverio. I hope the rest of the listeners here could relate to me, and the rest of the faculty of MS in Medical Technology. So this afternoon, I am tasked to elaborate more on the following. First, to identify the different points to consider in choosing your research topic. Identify the essential elements that must be included in your research title. And at the end of this discussion, somehow to find areas for medical science researches. During the pandemic time, we relaxed our requirements on our thesis. I'm speaking about our experience at San Juan de Dios Educational Foundation, Incorporated. So as the Dean of the College, we allowed descriptive researches to be capitalized where phenomenological researches surfaces or was utilized by the students. But we are now in the new normal, and we are going back to what we are expected of us as students of medical technology and graduate of MS in medical technology. So as a student of this discipline, we are expected to have researches on experimentation. So I will just give you some points about these experimental researches. So experimental researches or research are course of action is tried or carried out, and its effects are observed and measured objectively. So it's really pure research. However, in my group last time, I know merong mga working on thesis which are on a descriptive in nature, pero meron pa ding quantitative part on their descriptive researches. So remember that two groups are utilized for purposes of carrying out the effects. So we have to have our test group and our control group. According to Lack, Wales, and et al., they described experiment using the following characteristic. So the first one is the variable under test, or maybe we have to consider the treatment or solution hypothesized as being causal, can be applied deliberately, and its nature can be specified. Second characteristic is that the result or payoff from its application can be identified and measured in terms relevant to the given problem. And the third one, we have to identify the extraneous variables in the environment which is plausibly could affect the result materially, that also the materials that could be identified in our study. Next one, the test or experiment must be able to produce valid result, both internally and externally. So we have to make internal validity and external validity. When we say internal validity, so there is no other plausible cause of the observed results should exist except those that are being tested. And external validity, these are conclusions that can be projected to the real world problems. The researcher manipulates at least one independent variable, controls other relevant variables, and observes the effect on one or more dependent variables. Those independent variable is the experimental variable, which is the cause, while the dependent variable is the criterion variable, which is our effect. In our experimental study, we are guided with at least one hypothesis, which states what is our expectation or the causal relationship between the two affirmation variables. So we choose our group, we choose our subject. So we could randomly assign our group or our subjects. So we have also our control groups. However, there are, as mentioned earlier, there are several challenges that may come along during our experimentation. Okay, sa ating journey, sabi nga earlier, it was described that research is a journey. I agree 100%. And during that journey, there are challenges that you may encounter. Okay, number one, since we are on experimentation, we have to consider the cost. Okay, so there is really a cost for experimentation. Kaya I'm suggesting to those students na medyo mag-ipon-ipon na po tayo. Next one, it is time-consuming. And it requires a host of materials, supplies, and equipment. And since this is an experimentation, we have to look for a research laboratory that would carry our experimentation. Why is it we as a student of MS in medical technology and students of medical technology, requires us to do experimental research, because when doing our experimental research, we can be better assured of our study's validity, reliability, and accuracy of our study or experimentation. So you can ask yourself, are you ready? Okay, are you ready to handle all the challenges during your journey in the completion of your research paper? So you may either have a yes and no answer. But if you have decided to have a yes answer, okay, it's time for you to do your preliminary research. You can narrow down your topic, you can refine it, then develop your topic. You can state the topic in a form of specific statement or question. Then you identify the main concepts, terms, and keywords that describe the topic. But don't be disheartened if your response is no, because you have the time for you to read, and be prepared when the time comes that you are ready to say yes, because doing the research is a commitment. Okay, so if you have refined your topic and your heart was put into it, okay, it's now time for you to have your research title. So what is a research title? The research title shows what the study is all about. And this is probably the most important single part or the single decision you have to make in doing your research. So it serves the frame of reference for the entire study. It enables the researcher to claim ownership of the study, and it can help other researchers refer to the work for possible theories related to their proposed studies. If your research title is very clear in your mind, therefore, your way to the completion of your research is also clear to you. But we have to consider what are the essential elements in our research title. Okay, so we have to include the following. So we have the independent variable, we have the dependent variable. Sometimes we have to include in our title words such as effectiveness if we are trying to experiment on the effectivity, for example, of a, let's say if we're experimenting about certain plant and we use it in certain bacteria or any similar term to that, or maybe the word relationship if we're trying to compare two variables or any similar term. We have also to include respondents in the study if it is very relevant, the research venue if it is very relevant also, the time frame. However, we have to consider that our title must be very brief but concise. Should not be more than 15 substantive words, excluding the function words, which includes the articles, conjunctions, and prepositions. please bear in mind that, okay, kasi meron tayong knowledge or ang akala natin, the longer the title that we have, the better. So the longer the title, parang mas medyo yung research title ay medyo mabigat. Okay? So oh wow, ang haba ng title. Okay, but anyhow, just to give you the idea, when I proposed my thesis in MS in medical technology, my first title was Determination of Hypercholesterolemia Using Saliva with Nanoparticle-Induced Reagent. Okay, so that's my title. Okay, as I do my experimentation, and after the proposal and the second proposal or the second presentation, and up to the last presentation, the panel have refined my title, okay, into final title, which is Diagnosing Dyslipidemia Using Saliva. Okay. So, ilang words lang po yan? 1, 2, 3, 4. Okay. 4 lang. Sabi ko, bakit 4 lang po? Since the heart of the experimentation is about the nanoparticle, why don't we include this one? But I have to abide with the panel because they are the best person to suggest on what would be the final title. And I 100% agree with them because there might be some surprises as how was able to determine the dyslipidemia using saliva. So as they read on the paper, they will discover the inclusion of this nanoparticle. Okay? So you have lesson for that. Always abide with your panel. Okay. The first part really of having your research is really to think your topic, okay, before your title, okay? So, sabi nga napakahirap gumawa ng title or mag-isip ng problema bago ka sa research. Yun talaga yung pagbubuhusan mo ng oras. Kasi kung hindi ka makaisip ng tamang topic, ang mangyayari sa atin ay time will come, mawawala yung attention mo, mawawala yung, kumbaga meron kang angst o yung gigil ba, na gustong-gusto mo to. Tapos in the long run, magsasawa ka. Okay. However, we have to make sure that the topics that we want to work on are those topics that we love to work on or we are very keen in working on it. So what are the topics that may be suggested to you? Okay. So number 1 would be problems confronted in your laboratory work. Okay. Take time to look for possible topic in your present laboratory work. Nandyan lang po siya talaga siya. Or maybe in the process of the delivery of service in your workplace. Okay, I have read on one research topic which he deals about the ergonomics, okay, the ergonomics of the place he is working. Okay? So, ang ganda nung papel. So, if you think very descriptive siya, pero napakaganda ng execution ng kaniyang ng papel, and the problems, the SOP that was given was very good. Okay? It may be about the management. Okay? So, meron ba kayong gustong palitan na process, hindi yung manager, ha? Okay, yung process sa pinagtatrabahuhan nyo, how the laboratory is being managed, the protocols, the existing, baka naman may mas effective protocols kayong naiisip. Why don't you experiment on that? And also the procedures. Okay, when the nanoparticle was conceived, okay, I was really keen working on clinical chemistry, and nanoparticles was increased in attention that time. So I have used your saliva as the experimental substance, or tawag dito, sample rather, yung sample ko, para kasi makaiwas tayo sa mga tawag dito, yung mga process na kung saan ay masasaktan yung tao. Okay, so baka pwedeng yung saliva, kukuha lang, then after which, makukuha mo na kagad yung yung cholesterol level. Okay? But the first question is, paano tayo kukuha ng nanoparticle? So we need to find a laboratory wherein I could get a silver nanoparticle. Okay, it's good that may laboratory. I think the laboratory of Emilio Aguinaldo in Cavite, so they have the facilities there. So, nag-extract kami ng nanoparticles. Then, after extraction with nanoparticles, pina-analyze ko to sa RITM. Pina-check ko if nanoparticle talaga yung nakuha ko. Then, after which, I compared the result of yung saliva with the actual enzyme used to detect your cholesterol. Then I put a drop of nanoparticle to the reagent. Okay, then the same sample, so they were analyzed together. Okay, so just to give you a brief so nag-increase talaga yung determination ng saliva, cholesterol saliva using the reagent with your nanoparticle. However, the recommendation at the end is what would be the normal value for your saliva, okay, if there is no nanoparticle in it. Okay? Okay, so that's my experience. Okay. So it could be something that you can really work on in the laboratory. It could be something in the procedures that we are working on. Okay. So sometimes kasi naiisip natin, medyo na-delineate na tayo dun sa natin. Ano ba yung ini-expect sa atin as medical technologists? Okay. Sometimes we really work on yung mga susceptibility testing, tinetest natin yung mga plants. Sabi nga ng mga pharmaceutical students, parang nananakawan natin sila ng kanilang ng task, because that is their task. Okay, but anyhow, okay, just to give you an idea, sa school namin, as much as possible, dinidiscourage na namin yung mga ganong mga thesis. Okay, yung nature ng thesis na gano'n. Okay, so it could be technological changes. Okay, and curricular development. This one may sound descriptive in nature. However, your advisor can help you journey with these curricular developments. Okay, my doctor in education works on curricular development in medical technology. Okay, but however, that is a descriptive in nature. But on master's level, this could also be considered, lalo na nag-change na yung ating curricular offering. Ito pa yung isa na very interesting. We are very challenged to work on diagnostic kits. Kasi parang tingin ko, ito yung hinihingi sa atin. Okay, we have to make something that we will really work in the laboratory. Okay, so you think of diagnostic kits that would really determine the present condition of the patient, okay, and also development of biomarkers. One panelist that I have asked when the topic was given to me by Dr. Sherwin, I asked her, Doktora, ano bang pwede nating ipa-research sa mga student natin? Sabi niya, you know what? Parang yung researches natin would be parang, okay, yung nature niya ay nalalayo na talaga to what is expected of medical technology graduate. Okay, so we have to develop certain biomarkers. Okay, we have to develop certain biomarkers that would really address yung condition ng mga patient natin na bago siya mag full-blown. Like for example, creatinine. Creatinine could very good marker for kidney function test. However, ang creatinine talagang nag-i-increase lang pag meron na talagang widespread destruction ng kidney natin. Okay, so may mga biomarkers tayo na discover din ng ibang researcher. Okay, so yun yung hinahanap sa atin ngayon, na mag-invest tayo, mag-develop ng mga biomarkers. Next would be, okay, ito naman is I have asked again another friend of mine, Sir Ricky Martinez. Okay. Sabi ko, Doc, Sir, ano po ba ang pwede nating isuggest sa mga nagre-research sa atin about blood bank, sabi niya. So this could be about, sabi niya, you can identify different antigen, antibodies, specifically among Filipinos. Sa book natin ang libro natin na ginagamit sa blood banking, napakaraming antigen and antibodies doon. So, kumbaga, yung antigen and antibodies doon ay sa mga foreign population. But ano ang antigen and antibodies that could be inherently found among the Filipinos? Okay. And also, he suggests, so we have blood typing. So this is blood typing or maybe RH typing among ethnic groups, among the Lumads, the Bajaus, and discover something different in their blood types. Okay? So these are their suggestions. Another one would be the use of other specimen. Okay, that is not routine. Like for example, lagi tayong blood. Okay, so yung mga hindi masyadong nasasaktan yung mga patient natin. Okay, like for example, I have used your saliva as in determination of dyslipidemia. Okay, another one is we have to consider the agenda, okay, of the National Unified Health Research. Okay, so I will give you the NURA or the National Unified Health Research Agenda in 2017 and 2022. So in this kind of researches, you can get funding to this one. And these are the research agenda. Those that, one that would be responsive to the health system, a research that would really enhance and extend healthy lives, research on holistic approaches to health and wellness, research on health resiliency, research on global competitiveness and innovation in health, and research in equity and health. Each of these 6 themes has a corresponding research areas and description, which aim to guide the readers in determining the kinds of research that may fall under each specific theme. A research to enhance and extend healthy lives to produce evidence towards the prevention of disease, restoration of well-being, and reduction of disease burden. Our research must be focused on how we maintain among Filipinos happy, healthy, wealthy, productive, and sustainable health. On adolescent health, so these are the key issues on adolescent health. So we have teenage pregnancy, mortality from teenage pregnancy, high-risk behaviors, mental health, HIV/AIDS, and their corresponding policy services. Kagabi habang nag-iisip ako, so napakaraming teenage pregnancy right now. And sabi ko, is there ang kanila bang glucose level, okay, ang glucose level among teenage mom would be comparable sa glucose level ng iba-ibang age group. Okay? So, di ba meron tayong tinatawag na gestational diabetes? So, pare-parehas ba sila once they are pregnant? Okay, pare-parehas ba na may gestational diabetes? Or meron bang certain age group yung ating gestational diabetes? All we know is that when you are pregnant, you may be susceptible to gestational diabetes. Okay? Then, as to communicable diseases, so research on diagnosis, treatment, and prevention of eto mga re-emerging na to dengue, diarrhea, HIV/AIDS, and other sexually transmitted diseases, tuberculosis, rabies, neglected tropical diseases, and soil-transmitted helminthiasis, and other communicable diseases. Okay? So, eto pa rin yung ating problem, and they are re-emerging. So, you can work on this area, okay? You can work on this area, or you can consider a topic on this area. You can consider to work on environmental health, such as research on the exposure and toxicity and other effects of poor environment conditions and on health, including water quality and management. So very interesting tong water quality na to. Okay, so there are issues right now of water supply in Metro Manila. The question is that clean ba yung mga water reservoir natin? Okay, so if you are working on water, water treatment facility, I think this is a very good topic for you. Clean ambient and indoor air, solid and healthcare waste management. Akala natin, hindi pang medtech to. Okay. So you can identify what are those microorganisms that you can found in the healthcare waste, or maybe yung mga waste natin sa mga bahay, ano yung pwede nating mga makuha dyan. About sanitation and hygiene, and integration of health in environmental and urban planning. As to maternal, newborn, and child health, so studies on factors influencing compliance, service utilization, management, and effectiveness of interventions directed towards the improvement of maternal, newborn, and also child health. For non-communicable diseases, so research on early detection, prevention, and control of non-communicable diseases, such as cardiovascular diseases, cancer, diabetes, chronic renal diseases, risk factors, and evaluation of interventions and programs. So you can think of biomarkers, most probably, that could address these non-communicable diseases. And we think on how we can determine the susceptibility of a person to this risk. As to the diagnostic, we have to really go on development of diagnostic tests and devices for early detection and monitoring of pre-morbid condition and disease. So para talagang we are being called to really dwell on early detection. Okay, so we think of how can we improve the existing tests in the laboratory, or how we can add on to the existing test and use for early detection of certain diseases. For drug discovery and development, research on the identification and standardization of natural products and the development of drugs and vaccines for specific conditions. For example, yung cancer, skin diseases, dengue, and Zika, and multi-drug resistant infections for production, local use, and international marketability. Kung may mga estudyante dito from the RITM, napakadaming research study that you can work on in your setup, in your work setup. Napakaraming pwede nating gawin. Then information and communication technologies for health research on data science and e-health solutions for the integration of health information in recording and utilizing patient information, mapping of health risk, health resources, telemedicine, and health promotion. For biomedical products and engineering, so we are asked to research on improvisation and local development of reliable, safe, and affordable biomedical devices for supportive and therapeutic care for local health service provision and international markets. Okay, so yung mga miniatures na, because biomedical yan, but we could ask help from these biomedical people on how we can improve what we have in the laboratory. Okay? So yung ating point of care testing, baka in the long run, you can discover a point of care testing. Okay? So kung meron tayong point of care machine, baka naman yung strip yung ma-discover natin. We can never tell talaga kung ano yung pwede nating magawa. Okay? So this is about omic technologies or generation of new knowledge about health and disease using your genomics, proteomics, transcriptomics, and metabolomics. Okay? And for geriatric care, ayan, this is another area that is very interesting. Research to map the health needs of our geriatric patient kasi there is a specific needs, a specific requirements in terms of testing among our geriatric patient. The values that we have, for me isa yun lang ang pananaw ko, the values that we have existing in the laboratory would suggest healthy populations or adult populations. Healthy populations and adult populations. So kung hindi sumakto doon, so ibig sabihin, bagsak na yung geriatric natin. Okay, baka naman may ibang protocols or values among healthy geriatric ang pwede nating i-follow up or pwede nating maging standards. Okay? So, okay, another one would be appropriate health programs for them, policy, services for our geriatric population. In your existing work premises, okay, what is your SOP in terms of geriatric? O syempre, alam naman natin na pagka matanda, meron siyang may courtesy leave. Okay? Ano pa yung ibang services natin na pwede nating i-include among our geriatric patient na mag-i-improve ng ating services? Okay? So alam natin pag nag-improve yung services mo, word of mouth yan. And that is improve also the condition of the medical technologies in that clinic. Okay? Then also in HIV, AIDS, and other sexually transmitted diseases, so social behavioral studies to address the stigma, discrimination, and other barriers to HIV and STI screening and control. Okay? So, these are just some of the ideas or possible areas that you can work on. Okay? And don't be disheartened kung wala man dito yung gusto nyo, but anyhow, ang importante lang is that when you think of your topic, when you think of something that you will really work on, you have to be focused on it. Okay? As mentioned earlier, it is a long journey, and the journey may be hard sometimes. Okay? And it's not going to be easy. Okay. However, I am telling you what is very sure is that it's going to be worth it. Okay? Thank you and good luck to everybody for your future endeavor. Thank you so much for this opportunity. Good afternoon.
Thank you so much, Dr. Rosana Tala, for that very enlightening discussion. So a reminder to everyone, we shall see our guest speaker later in the open forum. So to our viewers, if you have questions, kindly type it in our chat box and we shall ask them to our speakers and thesis presenters later.
To introduce our next speaker, the next plenary speaker is the program chair of the PWU School of Medical Technology. He obtained his degrees in Master of Science in Medical Technology and Doctor of Philosophy in Education, major in Educational Management from the Philippine Women's University. He earned his degree in Bachelor of Science in Medical Technology at the University of Negros Occidental, Recoletos, Bacolod, where he graduated magna cum laude and the recipient of the PAMIT Safeguard MedTech ng Kinabukasan National Scholarship Grant. His research studies include overseeing limited in-person classes in medical technology, reference to a post-pandemic supervisory plan, and the correlation between the learning styles, online tools of the academic performance of medical technology students, implications to curriculum and instruction, detection and morphologic differentiation of plasticities in fecal specimens, and lastly, microfilariae in soil-transmitted helminths prevalence, among others. Let us all welcome to enlighten us in minding the research gaps for thesis completion, Dr. Sherwin B. Torian.
Good afternoon, and thank you very much for that introduction. I hope this one's already visible.
Yes, sir.
Now, the topic is about minding the research gaps for thesis completion. Now, the purpose of conducting research is that we have to close an existing research gap by providing a new body of knowledge, which is why it is important for us researchers to identify research gaps in our area of interest. Now, research attempts to contribute new scientific research literature rather than simply duplicating what already exists. Now, in the School of Medical Technology, we require that our students perform or complete a thesis which is unique and should be contributive to the body of knowledge in medical laboratory science. First, let us define what is a research gap. Now, a gap is something that remains to be done or learned in an area of research. It is a gap in the knowledge of the researcher in the field of research, or an unexplored aspect in one's area of study, that which is still not answered by research. Now, a more comprehensive definition of the research gap is offered to us by Robinson et al. And according to them, a research gap is that body of knowledge that may still be lacking, undiscovered, not explored, and void, or leaving a vacuum, which therefore raises certain questions to be answered by yet another research above what is already known concerning a phenomenon. So a lot of researches have been done on one particular topic. However, the researchers may have various approaches on doing a particular research. Now, a research gap can exist even when there's already a fair deal of existing research about a topic. But where the findings of the research pulls in different directions, making it difficult for you to draw certain conclusions, that is when there would be conflict of conclusions. Now, note that certain problems encountered may not be considered as research gaps. That is why, for example, you lack the needed materials which can be procured. That is not a research gap. If you are not familiar with the methods on how to proceed, but there are existing means to do it, that is not a research gap. If you don't know how to purify or cultivate a microbial culture, but literatures have suggestions on how to do it, or if you lack the literature sources for review, there are adjacent studies wherein you could find the sources for your RRL. Now, these kinds of stumbling blocks are not research gaps, but just setbacks in thesis research progress that we can find the ways to remedy. To proceed, let us define what are the types of research gaps. Firstly, we have the evidence gap. Now, an evidence gap occurs if a new research finding contradicts widely accepted conclusions, classical results. So it occurs if results from studies, newer studies, allow for conclusions in their own right, but are contradictory when examined from a different point of view. For example, several classical studies show us that, or tells us that blastocystis hominis is just commensal. However, newer studies now cite that blastocystis hominis is already pathologic. So the findings comparing the newer studies, findings comparing that to the previous and classical findings would be contradictory and maybe even inconclusive. That is why more and more studies are even made in order to really settle the score on what would really be the status of a special parasite such as blastocystis hominis, whether is it really commensal or it could be high up in the pathogenicity list. Now, the identification of contradictory evidence starts with analyzing the references first. When you do research, you gather a lot of references, especially for your RRL. The results of these analyses need to be presented in the synthesis of your chapter 2 of your thesis in order for you to reveal the contradictory evidences. Now, the next type of research gap is your contextual gap. Now, I agree with Dr. Rose. A contextual gap exists when there's already a decent body of existing research on a particular topic, but, however, an absence of research on specific contexts. For example, we have normal values or reference points of blood chemistries for Caucasians, for Europeans, but do we have normal values for Asians? No. Do we have normal values for Filipinos? No. Why not? Because no one is doing the research for that one. Maybe it is waiting for you to do the research so that we would be having our own Filipino normal values. Now, in a contextual gap, there would be studies about certain populations, an encompassing result about certain populations. However, specific groups are not being studied. The result of specific sample group may not be similar to the result of a general population. For example, a specific population has been studied, but perhaps a certain age group's result or a certain gender from that particular population or a certain ethnicity from the general population's result may be different from that of the result of the totality. Or even in a geographical area, for example, there may be studies in North America, there may be studies in South America, but there is no study for a specific country, a specific region, or even just a city. Now, the contextual gap is a popular option for dissertations and theses, especially for the first-timers. Why? Because, for example, there's already a study about Asians for their blood sugar. Now, you want to know what would be the levels of blood sugar for Filipinos living in cosmopolitan areas. So you just adapt the methodology of the study conducted on Asians, and you do this in a specific a city, just like Manila for Filipinos. So you already have the conceptual framework. You already have the protocols and procedures to follow. You are not doing it among Asians or a certain group of people on a wider scale, but you are narrowing it down by performing it among Filipinos, particularly in a specific city. So take advantage of that particular research gap, which is the contextual gap, by making use of very specific studies now patterned on a general study that was already conducted. The next type of research gap is what we call methodological gap. Now, this type of research gap emerges as a result of using repeatedly the same research methodology or design. Now, the methodology of existing studies may be lacking a certain perspective. Now, because you are using repeatedly this particular method. Now, for example, the bulk of existing research in bacteriology has taken a quantitative approach. It is always empirical. But why not look on doing bacteriological work qualitatively? There is a lack of rich insights and even texture that a qualitative study could provide. Also, in the detection of fecal parasites, we always make use of the same methods, direct fecal smear, if not direct fecal smear, the concentration method. So why not use a different method in doing the research? Why not capitalize on staining techniques? So when you are working on your research, you tend to study and base the concept on your readings, from your readings. Now, usually there is a commonality when it comes to the methods that are being used. Now, you feel safe that when you are actually doing the same method as what the other researchers did, you feel safe about it. But why not venture in a different method and let's see what you have? The next gap is what we call theoretical gap. If one topic is being explained using various theoretical models, there might be a theoretical concept. Okay, so for example, you are studying about heart diseases. Okay, heart diseases, there are two very popular theories about heart diseases. And these two theories about heart diseases are also conflicting. What are these theories about heart diseases? The first theory is the fat theory, and the second theory would be the sugar theory. The first theory is the fat theory, wherein when you have elevated cholesterol and triglyceride, of course it would burden your heart, causing heart diseases. But the second theory, which is a conflicting theory, would be the sugar theory. According to the sugar theory, persons who have normal pancreas, however, when they eat much carbohydrate-rich food, it would stimulate the production of more and more insulin from their pancreas. The effect of an elevated level of insulin would thicken your blood vessels. It would harden your blood vessels. It would inflame your blood vessels. And in turn, your blood vessels would be less flexible. If your blood vessels would be less flexible, it would stress out your heart. So these are the two theories about heart diseases, and they are actually conflicting. So theoretical gaps are a common occurrence in examining prior research on a subject. So when you are doing your RRL, you may find it confusing. Ano ba ang ilalagay ko sa RRL ko, or what theory should I be following? Now, our research framework can either adopt a conceptual framework or a theoretical framework. If you are adopting a theoretical framework, you are utilizing a known, established, and validated theory in dealing with your research topic. However, if you would be making use of conceptual framework, you are using something which is a roadmap, or you are creating a roadmap for your study, helping you visualize your research project and put it into action. So it is your choice whether you would adopt a certain theory for your theoretical framework, or you would rather adopt a conceptual framework. Now, the theoretical gap will arise if the researcher chooses an alternative theory to explain and proceed with the study. So why not? If there are two existing theories, the sugar theory and of course the lipid or the fat theory, maybe you could find something else. Hindi kailangan dalawa lang yan. Maybe you could find some theory to also explain heart diseases, not the usual sugar or lipid theory. The next research gap is what we call the knowledge gap. The knowledge gap is what is that which needs to be filled by new research, either because we know little or know nothing about it. Now, for example, in molecular biology, now we don't know how to create complementary DNA in the absence of reverse transcriptase. Is there another way to creating complementary DNA aside from making use of reverse transcriptase? Or why are we creating a reverse transcript, or why are we creating complementary DNA? Why not create complementary RNA instead? Because our template is already RNA. So there are actually other areas in doing our research that would actually offer or reveal a lot of knowledge gaps aside from molecular biology. Secondly, another example would be in dealing with mycoplasmas. The problem with mycoplasma, if you are working on mycoplasma, is that it would be very difficult for you to purify mycoplasma if you would be introducing mycoplasma as a pure culture to inoculate it on rats. So that would be a knowledge gap. So what you do is you create your own technique. You create your own theory in order for you to be able to bridge the gap, and in order for you to complete your thesis. So find ways in order for you to do the purification, find ways in order for you to create complementary DNA or complementary RNA without the use of a reverse transcriptase, if that is possible. Now, there are two settings where a knowledge gap might occur. First, the knowledge may not exist in the actual field from related research domains. There's nothing in the RRL that you could find. So what do? You are stuck in your research because there's nothing in the RRL that you could find about it. So you would be asking yourself whether should I be continuing with the research, or should I be changing my research topic if I cannot continue with this present research because of the great void or the great knowledge gap. Secondly, results of a study differ from what was expected or does not offer an answer. I am expecting that this particular inoculum would cause pathogenicity to the mice. I am expecting that it would destroy the small intestines or the microvilli of the mice. But upon doing the preliminary investigation or the preliminary trials, there was no effect at all. So what should I do? So what went wrong? So the outcome is different from your expectations. There is a gap there. Now, most importantly, we should be asking ourselves, how is research gap important in a research thesis? Now, we should be taking advantage of the research gaps that we encounter, and we should be using the research gaps positively in our researches. How should we use research gaps in our research thesis? First, the research gap can actually serve as a topic of study. Now, finding a research gap in a topic or in a subject upon your readings gives you an opportunity to investigate and to form a concept in order to pursue in a research thesis. So you are asking yourselves, what would be a good research topic? You have no idea. But upon reading, you stumbled upon an article, and your own observations and your own insights tells you that there is something lacking about this research. Once you have already identified that research gap, you could actually make use of that research gap as your topic for research. The research gap can set a study in a unique direction. So when all the other studies look at a population in general and find meaning in the generalization, a researcher may focus on one specific area or group or the same total population. When all the researchers are using the same approach in its investigation, a newer and more innovative way may be established in order to offer a different perspective. So do not be intimidated when you are doing your research. Do not try to follow the safe path on research. You could always find something which would make your research unique, and then try to venture on a different direction from that of what the majority of researchers have already ventured on. So again, one of the characteristics of our research thesis for School of Medical Technology is that we want to make it unique. Maybe not only us, but also in other programs as well. We all want our thesis to be unique. The research gap allows you to identify concerns to annotate. Now, in analyzing the research gap, the researcher has specific areas of the reference articles to annotate. And in doing the annotated bibliography, you're already gathering related literature for the chapter 2. Now, in MS, MT, and MS HSM, your requirements for the comprehensive examination is your annotated bibliography. And in choosing your annotated bibliography, you are to choose a topic. If you have chosen a topic which is also the topic for your research thesis, then you are already doing your RRL, or the review of literatures. Now, in annotating those articles, you may stumble on the research gaps. And it is very important that you share your insights about the research gaps. Now, rather than simply summarizing the articles that you have read and making it or submitting it for your annotated bibliographies. Also, the research gap allows a researcher to classify the related literature. Now, noting the various gaps will allow the researcher to group the articles and studies that have been gathered for review. Now, this particular article is or can be grouped because of the common research gaps among them. And these articles can be grouped because it tells positively about a certain topic that you are dealing with. These articles are grouped because they go contrary to what you are expecting as a result in your research thesis. So this can be reflected well in the synthesis of your RRL. And finally, the research gap can inspire a new approach to explore a topic. Methodological or even theoretical gaps may motivate a researcher to come up with different approaches in pursuing a not so new topic, and it would give it insights. It would be you would be giving fresher insights in understanding a particular subject. Now, we should perceive research gaps as opportunities for further scientific exploration, which is critical for improving future theoretical insights in the different scientific disciplines. Hence, the significance for researchers is to provide implications for future research. And as men of science, we should identify and use the research gap to our advantage. And identifying a research gap is one of the crucial parts of many research projects. And according to one researcher, spotting a research or a knowledge gap and setting out to address it is generally what makes your research worthwhile. So make use of your research gap positively, capitalize on it, and turn it into a worthwhile research study. Now, these are my references. And thank you very much for your undivided attention.
Thank you, Dr. Sherwin Sariana, for completing the loop in the preparation of the thesis title and understanding the gaps for its clear completion. At this juncture, let us watch the activities that made us think and enjoy in the year that transpired. After the plenary session with our speakers, let us now proceed with the presentation of our students who have completed their research thesis. So for our first presenter is our very own college lecturer and clinical instructor, fresh from completing her master's thesis. Let us all welcome Miss Irish Ponce.
Hello, good afternoon to each and everyone. I am Irish S. Ponce, and I will be presenting my research study, which is extended release niacin effect on electrolytes and liver enzymes of alloxan-induced male Sprague-Dawley rat. So, this study is a good example of Dr. Sherwin gaps. Example of these gaps, it's about the knowledge gap. So the knowledge gap in my study is that this research focuses on the extended release niacin effect on diabetic male Sprague-Dawley rats, and after 5 hours of introduction of the niacin or your extended release niacin to the subjects, the electrolytes and liver enzymes were tested using 2 assays, which were the ISE or your ion selective electrode test, and then the colorimetric method. So when I say ISE, this is the determination of an analyte wherein by the convention of the activity of a specific ion on a dissolved solution into electrical potential that is detected. And for your colorimetric method, this is the detection of the concentration of a specific analyte through color production. So this study findings showed the effect of the drug to the kidney and liver. So the extended release niacin is known to be a good supplement in lowering the lipid profile of patients. However, it didn't specify in those studies if they have the effect on the electrolytes and the liver enzymes or the effect of the ER niacin to diabetic patients. So that is why I introduced the aloxan to increase blood sugar of the patient. Now let us move on to the statement of the problem of my research study. So generally, this study aims to determine the effects of extended release niacin and electrolytes like sodium, potassium, chloride, and liver enzymes like your alanine aminotransferase, aspartate aminotransferase, and your alkaline phosphatase of aloxan-induced diabetic male Sprague-Dawley. So this is study specifically seek the answers of the following questions. So for the first one, what are the baseline levels of the electrolytes, which I said before, using the ISE and colorimetric assay. Then for the liver enzymes, these three, your ALT, AST, and ALT, using the colorimetric assay of aloxan induced male Sprague-Dawley. For number two, what are the levels of the electrolytes and the liver enzymes using the two assays of using the varying doses or milligrams of extended release niacin, which are 500 milligrams, or 750 milligrams, and 1,000 milligrams. So these milligrams are according to the cost pharmaceuticals. So for the third one, is there a significant difference? So we are now in the test of significant difference between the baseline of the electrolytes using ISE and colorimetric method and liver enzymes using the colorimetric method only, using varying doses of extended release niacin. Then for the fourth one, test of significant difference of the varying doses of extended release niacin to the control. And for the varying doses of extended release niacin to the different treatments. And then for number 5, is there a significant difference in the sodium, potassium, chloride, and liver enzymes like the ALT, AST, and ALT levels as compared to the control? So the control is the distilled water. So for the sixth one and the last one, which method can be proposed to detect the effect of extended release niacin on different analytes, whether it is the ISE or your colorimetric assay? So we are now going to the methodology. So this is the materials and subjects. So in this picture, we can see the materials like syringe, tube, disinfectant, and the pure agent, which is the aloxan, this one, and the niacin. So this Vibrac, or also known as your Zolethil, is the anesthesia to be given to the subject before the intracardiac blood collection. It is Dorminal, or also known as your phenobarbital. This is for euthanasia. So the subjects were grouped into 4, and each group has 6 rats within the groups. So in here, we can see that this is the illustration of the baseline glucose testing, wherein before the introduction of the niacin, we should first test the baseline glucose of the rat so that we can introduce the aloxan. So this is the preparation of aloxan, and this is the induction of aloxan through intraperitoneal injection. So after that, 24 hours later, we will test again the glucose levels of the rat. So if they have greater than 180 milligrams per DL of glucose, they are included in the testing and baseline levels testing of analytes. So in here, we can see that this is a tail vein collection of blood of the baseline levels of analytes like your sodium potassium chloride and then the liver enzyme. After testing them for the baseline, we are now going to prepare the extended release niacin solution to be given to them through oral gavage. So this is the oral gavage of the CR niacin. So there are 500, 700, and 1,000 milligrams. So after giving them the extended release niacin with varying milligrams, after 5 hours, they will the subjects will be going to be extracted through the intracardiac injection. So this is the cardiac puncture. So it is directly to their heart. That is why we use euthanasia. So after the cardiac puncture, euthanasia will be given, or the phenobarbital will be given to them. And then to confirm if they are truly deceased, we confirm for the heartbeat, the breathing, and then the color of the subject. After that, the collected blood will be sent to the laboratory for testing. So there will be 2 assays, once again, the ISE and the colorimetric assay. So we are now here in the table, the subjects and designation of those. So for your treatment 1 or T1 control, which is distilled water, for number 2, the 500 milligram per kilogram CR niacin or your extended niacin. For treatment 3, the 750, and for treatment 4, the 1,000 milligram per kilogram. So this table here, the normal values, this is my basis for the normal values of the analyte, which is from the study of Len E.L. et al. From the reference values for young normal Sprague-Dawley rats. This is in 1996. So for table number 1, these are now the results. Table number 1 shows the baseline levels of electrolytes and liver enzymes using your ion selective electrode and colorimetric assay. So as we can observe, the sodium and the chloride for both techniques or assays are within normal limits. Also true with the liver enzymes, they are within normal ranges. However, as you can see, the potassium is lower. So they are in lower ranges. That is why the red color. So for table number 2, mean levels of electrolytes and liver enzymes after 5 hours. So after the introduction of the ER niacin. As we can see here, the sodium and chloride are still within normal ranges. However, for the potassium and the liver enzymes, they are increased. So the reason for this is that there is different, the reason for this one, this one, decreased levels of your potassium is because the alloxan is known to increase your glucose levels. However, the increased glucose levels also disrupt the levels of potassium. It shifts your potassium, your extracellular potassium inside the cells, kaya siya lower. Now, for the liver naman, for the liver enzymes, it is known that the alloxan causes acute liver damage and increased liver enzymes. This is according to your Thesis et al. 2015 and the Very Well Health in 2021. So now let's go to the test of significance of baseline levels of electrolytes and liver enzymes. As showed before, potassium for your ISE and colorimetric assays show significant difference between the treatment levels. You can see here from the table here, significant difference between the treatment and the control. So after 5 hours, nag-normalize na yung glucose levels ng control natin. So yung mga nasa ibang treatment, decrease pa rin yung potassium levels nila. So this is because yung niacin is known to increase the blood sugar. That is why there is a persisting decrease ng potassium level. So ALT and ALT shows that there is significant difference between the treatment and control. You can see here. So for the test of significance naman of electrolytes and after 5 hours of giving the extended release niacin, the sodium and chloride for both assays are within normal limits as showed before. And for the potassium levels, it remains decreased. It's a different treatment. However, for the control, it became normal. So it remained decreased. This is because of the niacin. Next, the findings of test in the test of significance of liver enzymes after 5 hours showed significant values of ALT levels for all treatments when compared to the control. Matataas lahat ng ALT levels. Also with your AST and ALT, no significant difference among the various treatments for the 3 treatments. However, if you compare it to the control, there is significant difference. Matataas lahat yung ALT. However, pag kinompare siya dun sa control, mas lalong tataas pa siya. That's why there is significant difference. So for the method, which method can be proposed? So both methods used in the measurement of electrolytes and liver enzymes are found to be efficient. There were no drastic changes. If you have seen it, point by point difference lang siya, even though they have different principles of testing, as I mentioned in the previous slide. They have different principles of testing, but they still have similar or precise na mga values. And for the conclusion, based on the findings of this study, the varying doses of your extended release niacin has no significant effect on the levels of electrolytes, like for your sodium and chloride using both assays, your IST and calorimetric assay, but for potassium, it remains lower. This means that there is no association of indication of kidney damage, but there is a disruption on the potassium levels because of the glucose after the use of extended release niacin. Now for the next one, the extended release niacin has significant effect sa lahat ng values of liver enzymes, which are the 3, the ALT, AST, and ALT. So this indicates now that the niacin has an effect on the liver and may lead to damage or hepatotoxicity. So that's it for my research study. Thank you very much.
Thank you, Ms. Ponce, for an informative presentation. Again, if you have questions, please type it in our chat corner and we shall be reading them later on. Next in line to present their thesis entitled COVID-19 Testing, Knowledge, Attitudes, Practices, and Preferences of Pateros Residents. Let us give warm of applause to our interns, Ms. Rhonda Caril Enriquez and Ms. Kimberly Marie Ibarra.
To our esteemed guests and our fellow students, a pleasant afternoon to all of you. Today we are here to present our research entitled COVID-19 Testing, Knowledge, Attitudes, Practices, and Preferences of Pateros Residents by Kimberly Marie Ibarra, Rona Caril Enriquez, Samantha Alcala, Paula Alessandra Alfaro, Kay Angeli de la Cruz, Arcy Erfe, Bianca Lynn Ordonez, Zaida Sofia Recto, and Ella Joy Verano. Diagnostic testing has been important to the global response to COVID-19. In the early months of the pandemic, particularly in the time when there was a lack of effective medicines or a vaccine, one of the few countermeasures available to governments to restrict the spread of the virus minimize transmission, and avoid fatalities was the quickly produced diagnostic tests. According to Bevan in his study in 2021, as the number of COVID-19 cases worldwide increased in early 2020, many public health experts recommended widespread rapid testing to supplement under other containment strategies such as hand washing, contact tracing, and quarantine. And that should be regarded as significant as face covering, social distancing, and vaccines. Countries such as China, South Korea, Germany, and the United States saw surges in late 2021, despite being pioneers and aggressive in their mass testing. The Philippines, on the other hand, suffered the same thing as a result of the liberal measures implemented over the Christmas holidays in December 2021. The IATF, in turn, boosted its precautions in response to the threat of another wave. Testing frequently implies unexpected costs, such as inconvenience and discomfort of accessing and undergoing testing, as well as the financial and social sacrifices demanded by self-isolation. As such, individuals at risk who refuse or evade testing may jeopardize a community's epidemic control and recovery initiatives. Public health researchers and policymakers must monitor public attitude and acceptability of testing in order to understand the underlying causes of their reservations. Such successful COVID-19 prevention and control is achieved through strengthening the general community's knowledge, attitudes, and practices. By being able to pinpoint their existing perceptions, beliefs, and practices, we can document the perceived barriers against COVID-19 testing. To the knowledge of the researchers, there has not been any local research that studies and has correlated the level of knowledge, attitudes, and practices of Filipinos towards COVID-19 testing, nor their preferred attributes and preferences towards the testing methods. Other research has focused on COVID-19 in general, but the current study focuses on COVID-19 testing methodologies and their respondents' health-seeking behavior toward getting tested.
Our study aimed to assess the knowledge, practices, attitude, and preferences of Pateros residents towards COVID-19 testing. So we, the researchers, sought to find the answers to the following questions. First, what is the demographic profile of the respondents in terms of age, gender, marital status, presence of comorbidities, education, and occupation? Second, what is the level of knowledge of the residents of Pateros towards COVID-19 testing? Third, what is the attitude of the residents of Pateros towards COVID-19 testing? Fourth, what are the practices of the residents of Pateros towards COVID-19 testing? Fifth, what is the preferred testing method of residents of Pateros towards COVID-19 testing? Sixth, is there a significant difference among the demographics with regards to level of knowledge, attitudes, practices, and preferred testing methods? 7. Is there any significant relationship between the level of knowledge and practices of residents of Pateros towards COVID-19 testing? And lastly, is there any significant relationship between the attitude and practices of residents of Pateros towards COVID-19 testing? This study used a quantitative research technique with online survey questionnaires with a hypothesis testing at 0.05 significance to see whether there are any significant differences across demographics in terms of knowledge, attitudes, and behaviors. Furthermore, we use a correlational study strategy to examine whether two quantifiable variables from the same group of respondents are significantly related. This study aimed to analyze any significant relationship between the level of knowledge and practice, as well the attitude and practice of Pateros residents towards COVID-19 testing. The respondents of our study were composed of 100 volunteer participants selected through convenience sampling and snowball sampling. The respondents ages between 18 to 80 years old and are residing in the selected barangays of Aguho, Aliw, Tabacalera, Santo Rosario Silangan, and Pagkakaisa. All of them have undergone COVID-19 testing and are able to read and comprehend the English language and have access to the internet and are capable of answering on Google Forms. The gathered data were tabulated, analyzed, and interpreted utilizing Microsoft Excel and SPSS for statistical analysis. As for results, Table 1, the gathered demographic are as follows. As we can see, age, majority of our respondents are within the age bracket of 18 to 33 years old. 55% of them are female, while 42% of them are male. As for the marital status, majority are single. As for the comorbidity status, 75% of our respondents are without comorbidity, while 25% of them are with comorbidity like hypertension or diabetes, and etc. Educational attainment, high school graduate are 32 of the respondents, while 43% of them are college graduate. As for the occupation, 62% of them are employed, while the other either have part-time jobs, student, or unemployed.
So on the level of knowledge of the respondents, a mean score of 8.01 was recorded and interpreted as above average rating as shown on Table 2. This table shows that the respondents were familiar with the fundamentals of COVID-19, such as transmission, means of testing, and detecting a positive case. Despite being above average, we can see that some people were unaware of the fundamentals and received a lower score, adding to the pool of this spectrum. This relates to the findings in the study of Sim and Embret in 2021 that misinformation regarding protective measures, transmission, and testing protocols is affected by health literacy in some people. At the same time, in the study of Gelbach et al., their study also expressed that some members of the communities might not have access to the internet, might not be proficient in English, to which most of the information regarding COVID-19 were written in, or have knowledge about, they don't have knowledge about how to access reliable public health sources for information regarding COVID-19. Instead, they might have been relying on the word of mouth, wherein definitions may have been lost in translation already, and or personal opinions have been incorporated into. If they were able to get this information from sources from the internet, it was possible that they were unable to discern or filter out credible sources from unreliable sources. Next, please. To determine the level of attitude and practices, Likert scale was used. The parameters are stated below to identify how much each respondent agrees with the presented statements on the questionnaire given. On average, they strongly agreed that early detection of COVID-19 virus could improve treatment and outcome, and that testing was crucial and essential for the country to eradicate the virus. This can be associated with their belief that the existing COVID-19 tests were accurate. With this attitude, it is expected that people from Pateros were more open to testing and contact tracing, evidenced by their strong willingness to be tested once they were exposed to someone who was known to be positive. Although some would be able to afford testing and isolation costs, it could still present a barrier into getting tested and isolated to some, as evidenced by the neutral rating. In the scoping review of Bevan et al. In 2021, they have seen the influence of socioeconomic status in the ability to get tested, wherein those who were only students, unemployed, underpaid, and have low income were unable to afford testing costs, even if they wanted to. Other financial burdens such as transportation costs, getting time off work for testing and for isolating when they become positive, and the isolation costs itself presented barriers for those in low socioeconomic categories. Next, please. So to determine health-seeking intentions of the respondents, questions regarding their practices when they were exposed to or positive were asked. The overall weighted mean response towards their practices was 4.414, which has a rating of strongly agree on the statements and indicates that they practice good habits as constantly advised by the DOH and LGUs. On average, the respondents got tested as soon as COVID-19 symptoms had manifested and already isolated themselves while waiting for the test results to be released. When they were tested positive, they did their due diligence of reporting their health status to the barangay or LGU COVID response units and their close contacts. Lastly, they have stated that they did not go outside throughout the duration of their isolation. These findings may denote the effectiveness of the protocols implemented by the local government unit of Pateros and the national government in controlling COVID-19 cases.
The reasons for the respondents to undergo COVID-19 testing, as we can see in the table, were primarily because it was required by work, having 33% of the response, and they were exposed to someone who is a confirmed case of COVID-19 patient. As for preference method of testing, their preferred method was RT-PCR, having 73%, and their preferred mode of testing is home-based testing, followed by in-clinic testing. Lastly, the attribute that they liked the most about their preferred method was its accuracy, and its least is its price. From these results, we can assume that accuracy and pricing were the attributes that are considered by the people when choosing the preferred testing method. As RT-PCR was chosen as the preferred method of testing and accuracy being its most liked attribute, this is an understandable relation since RT-PCR is considered the gold standard of diagnosis of COVID-19 and remains the most accurate method of testing. As for the price, as stated by Lee et al. 2020, RT-PCR is the most expensive option as compared to others like rapid antigen testing. Here's the significant difference among variables. Table 6 shows the significant difference in the CAP scores when grouped according to the respondents' profile characteristics. Results revealed that the knowledge scores were significantly different in terms of age, gender, presence of comorbidity, and education. Also, the attitude score showed a significant difference in age and education. The practices also showed a significant difference in marital status. The significant difference relates to the knowledge can be related to the finding of Rivero 2020, wherein he states that individuals who have higher education have greater ease in the use of information and communication technologies, and therefore they have more likely to search for information. The younger age brackets are considered the digital natives, and if we will compare them to older individuals, they grew up having the availability of gadgets. Also, middle to younger age brackets are keener in filtering facts compared to much older individuals. In the study by Al-Hanawi et al. 2020, it was discovered that gender had an impact since women had better practice and knowledge than men. But based on the data, the majority of respondents that have no comorbidity belong to the lower age bracket, and these young individuals have a higher rate of using information technologies. Thus, they have wider access to information. Having higher education can relate to the capability of an individual to comprehend the importance of testing, risk of COVID-19, and evaluating the consequences of not following certain protocols, such as isolation and undergoing COVID-19 testing once symptoms occur and once they have close contact with COVID-19 patient. Although it will require further studies, the significant difference related to practice can be related to the idea that married guardians are more concerned about the health of their family members, as this affects their responsibility in the family, such as providing financial support once one of the members got sick. Thus, they are more obedient in following COVID-19 protocols, such as undergoing COVID-19 testing. As for the relationship between the level of knowledge and practices, and practice score, the computed p-value is 1.539, which is less than the critical p-value of 1.98. And this means that there is a negligible correlation between the level of knowledge and practices. Thus, the null hypothesis failed to be rejected. A negligible association between residents' knowledge and COVID-19 preventative practices was discovered, in contrast to the favorable relationship between attitude and preventive practices. This result is at odds with that of earlier studies, which showed that having knowledge towards COVID-19 has a beneficial effect on preventive measures. The idea of optimistic bias, which arises when a person feels protected against risk, may have an impact on these discrepancies, though further research may be necessary for this kind of interaction. As for the relationship between the level of attitude and practices of the residents of Pateros is found significant with a p-value of 5.449, which is greater than the critical p-value, meaning there is a significant relationship among the two variables. Thus, the null hypothesis stating that there is no significant relationship between the attitude and practices of the residents of Pateros towards COVID-19 testing should be rejected, as there is evidence of a moderate positive correlation between attitude and practices of the residents. In addition, this is also interpreted that the Pateros residents have adopted good practices toward COVID-19 testing in relation to having a positive attitude towards COVID-19 testing. This result is similar to the finding of Tupal et al. In 2021, stating that if the general population has more positive attitudes, then they will highly advocate preventive measures. As for the conclusion, the study revealed good overall knowledge, positive attitude, and good practice of the Pateros residents towards COVID-19 testing. Respondents are aware of the importance of COVID-19 testing and when they should have one. Also, among the attributes, RT-PCR is the most preferred COVID method of testing by the respondents. In the assessment of correlation, it was revealed that there is a significant relationship between the attitude and practices of the respondents towards COVID-19 testing. And that is all for our presentation. Thank you for listening.
Thank you very much to our interns for today. And next would be our third thesis presentation, our Master of Science in Health Systems Management program. Ladies and gentlemen, please welcome Ms. Charmaine Grace Texon Balbin.
Good afternoon, everyone. Once again, I'm Charmaine Grace Vitexon. Today, I will present my thesis entitled Total Quality Management of Selected Blood Service Facilities in Metro Manila and Assessment. The aim of my study is that to determine whether the blood service facility is producing quality services and products while prioritizing the staff's welfare and the facility's upkeep, as well as the level of awareness of TQM practices during the period of pandemic. It is to provide recommendation for TQM enhancement based on the identified strengths and limitations of TQM within the blood service facility to reduce quality vulnerability now that the Manila alert levels have stabilized. For the statement of my problem, the study assessed the total quality management system of selected blood service facilities in Metro Manila as assessed by health professionals. Specifically, it answered the following problems. First is what is the profile of selected health professional at a blood service facility in Metro Manila in terms of gender, classification of a blood service facility, the designation, work schedule, employment status, and years of service. Next is how did the selected health professional assess a blood service facility based on the total quality management system standards such as leadership, resources and competency development, process management, teamwork and participation, and continuous improvement. Third is what was there a significant difference in the assessment of total quality management based on the profile of health professional in different blood service facilities? Fourth is based on the results of the study, what weaknesses and strengths were identified. And lastly, what plan was recommended for the improvement of total quality management? For the conceptual framework, it consists of 6 variables. The independent variables are of total quality management is leadership, resource and competency development, process management, teamwork and participation, and continuous improvement. The independent variable leadership refers to the standard that can influence total quality management by developing and implementing quality systems and improving the work environment by serving as a role model and increasing communication. Another independent variable is resource and competency development, which refers to a standard focused on upgrading and equipping resources and professionals to achieve total quality. The next variable is process management, which refers to the strategy of standardizing strategic policy and documentation in blood service facility and influences total quality management. Next is teamwork and participation. This is a variable that relates to a standard and can affect total quality management practice by changing how management and the health professionals and suppliers. Finally, continuous improvement refers to a standard that affects total quality management by focusing on facility performance measurements and feedback from people who receive services. And lastly, the dependent variable is the PQM improvement plan. This is a total quality management. In this figure refers to a company's strategy for ensuring the quality of its products and services. For the methodology, I utilize a descriptive survey. It is a study that employs quantitative methods to understand the assessment of health professionals on total quality management in their BSF. Next is for my respondents. Respondents were from the researcher. Respondents were from selected blood service facilities in Metro Manila. 60 health professionals with or without knowledge of PQM principles were randomly chosen to participate in the study. This includes a blood bank with additional functions, a blood station and blood collecting unit, and a blood center. To describe the differences of the blood service facilities as per Administrative Order number 208-008, a blood center is a blood service institution that can promote, recruit, collect, and provide health education and counseling to blood donors. It can also store, issue, and distribute whole blood and other blood banks, and test and process blood units for blood dispensing. Finally, it is in charge of blood collecting unit and blood station inside its blood service network. Next is the blood collecting unit and blood station. It performs the same purpose regarding blood donors. They collect and store blood products but not blood samples, which are tested and are processed in the blood center. While blood station can only encourage voluntary blood donation while storing, issuing, and distributing whole blood and other blood products. Finally, a blood bank with an additional function is known as a blood service facility serving the same purpose as a blood station. And they only differ in terms of the added function, which can include testing and collecting blood products or only testing them. Other functions can also be compatibility testing, direct Coombs test, red cell antibody testing, investigation of transfusion reaction, and assisting in hospital hemovigilance. Next is the sampling technique. I use a convenience sampling method, which is a non-probability sampling method where units are selected for inclusion in the sample because they are the easiest for me to access. Next is instrument. So to collect the respondents' views, I developed a Google Forms-based online questionnaire with two distinctive sections based on the review of literature. The researcher utilized a questionnaire because of its cost-effectiveness, accessibility, and respondents' confidentiality. The questionnaire was also tested for its validity using the Scale Context Validity Index. For the statistical treatment of data, the acquired data were tabulated and meticulously evaluated and carefully interpreted using the statistical methods. For the statement of the problem, number 1, the frequency distribution table was used for the participant profile. The mean was used for problem number 2 regarding the pandemic's effect on PQM standards. And for a statement of the problem 3, the ANOVA F-test was used to evaluate whether there was a significant difference in the assessment of individuals in total quality management. And the weighted mean was used to assess the results for problem 4 regarding flaws and strengths of total quality management of blood service facility. Next, for the data gathering procedures, the questionnaire was sent to the designated Metro Manila Blood Service Facility via Google Forms link, and data was collected between September 8 to September 29, 2022. For the profile, based on the responses, health professionals, among health professionals, females outnumbered the male health professional. Among the classifications of blood service facility, 43.3% of health professionals work in a blood center, 23.3% work in a blood collecting unit and blood station, 20.1% work in a 23.3% work in a blood collecting unit and blood station, 20.1% work in a blood station, and 13.3% work in a blood bank and other functions. For the designation, 65% are medical technologists, 18.3% are other health professional, and 11.7% are nurses, and lastly, 5% are quality assurance officers. Most of the health professionals surveyed have a work schedule of 8 hours, which accounts for 61.7%, 28.3% have a work schedule of 9 to 16 hours, 8.3% have other schedules besides 8 hours, and 17 to 24 hours, and 1.7% have a work schedule of 17.24 hours. For the employment status, 80% of the health professionals surveyed have a permanent employment status. 16.6% are volunteers and 1.7% are probationary or contractual employees. Lastly, for the years of service, a total of 40% of all respondents have 5 to 10 years of service experience. 35% have a 1 to 4 years of experience. 18.3% have 5 to 10 years of experience, and 6.7% have less than a year of experience. Regarding the assessment of health professionals, the mean score for continuous improvement is 4.36, process management is 4.26, and resource and competency development in total quality management on responses from health professionals is 4.24%. 4.24, which is considered very high. This mean score implies that health professionals have positively assessed their facility's total quality management system, standards regarding continuous improvement, process management, and resource and competency development. The mean score for leadership assessment is 4.11, and the mean score for the teamwork and participation aspect of total quality management is 3.99, which is lesser rating than the three standards. Nonetheless, this still suggests that the respondents' high evaluation of their facility's total quality management. In the test for significant difference in assessment, total quality management in a blood service facility among the profile of health professional, there was no significant difference in assessment of total quality management in terms of gender, classification of blood service facility, the classification of the blood service facility the respondents are working at, the work schedule, or employment status. However, the p-value for assessing the total quality management of a blood service facility based on health professional designation is 0.012, which is less than the p-value of 0.05. With this, the null hypothesis was rejected. This implies that there's a significant difference in assessing the total quality management of a blood service facility based on a resource and competency development, particularly on one's designation, in which we can see that there is a significant difference between the profiles of medical technologists and nurses, as well as between nurses' opinions and those of other health professionals, with the comparison between medical technologists and nurses being the most important. Lastly, a significant difference was observed in the assessment of health professionals on teamwork and participation, with the p-value for teamwork and participation concerning the years of experience being 0.028. This implies that the null hypothesis has been rejected. Therefore, inferring from your years of experience, there is a significant difference in how health professionals evaluate teamwork and participation in a blood service facility. A significant difference was seen between those who have worked in a service facility for 1 to 4 years and those who have worked for more than 10 years, as well as between those who have worked for 5 to 10 years and those who have worked for more than 10 years, which has the greatest difference exists between 1 to 4 years of work experience and more than 10 years of work experience. Based on the weighted mean of the standards of TQM, there is room for improvement in each of the dimensions, as these have a potential to influence total quality management. To summarize, teamwork and participation is the weakest TQM criterion from January 1, 2020 to June 30, 2022, requiring the most notable progress. This could be due to a need for more appreciation for superior quality work for health professionals and employees are neglected. One technique to make them feel valued is to express appreciation and reward them by team building, monthly meetings, and Employee of the Month program. Leadership is the next weakest. This could be because the focus of management is not on solving problems, but instead they find people to blame for them and do not prioritize mental health screenings. To further equip leaders in the BSF, a leadership and management training is a need, and as health professionals are vulnerable to mental health problems, necessitating interventions such as monitoring of their mental health to avoid harm to the health professionals. Continuous improvement, process management, and resource and competency development were among the most powerful indicators we examined. Resource and competency development is ranked third, as regardless of the circumstances, no unqualified, inexperienced, or ill-informed blood bank employees should be employed because they would be dealing with patients' lives and a single error could be fatal. So in order to maintain this competency development program is encouraged, and to add is resource audit and strategy upgrade to make sure that all resources are well used. Health professionals rank process management second. This could be because the total quality management training is provided regularly and incident reports are produced to aid in process improvement. Lastly, for continuous improvement, a TQM grade of very high. This could be because blood donors are given information and motivation to encourage them to donate blood regularly. But in order to encourage blood donors to donate regularly, an improvement of the process can be done by annual assessment of the process and their customer feedback to make sure that there is increased donors and patient safety and customer satisfaction. For the conclusion, women predominated in the interviewed blood service facilities in Metro Manila. Medical technologists dominated the respondents as they are the major workers required for a blood facility to function. The selected health professionals rated their blood service facilities' total quality management standards as satisfactory, as standards receive a rating of high and very high. According to the study's results, there is a substantial difference on how one's designations and years of experience affect how one's assess the total quality management of their blood service facility. According to the results, the least essential total quality management standard that the selected blood service facilities that were interviewed was teamwork and participation. And the most essential total quality management standard was continuous improvement. For the recommendation, the study showed that teamwork and participation were the most underemphasized total quality management standards in Metro Manila Blood Service Facility. However, all other measures of the total quality management standard can continuously improve as they have yet to receive the highest possible score. And future researchers may target other types of blood service facilities outside of Metro Manila and a larger number of respondents to provide more information and diversity. In addition, future research may solicit additional responses from customers and donors to learn more about the current state of TQM at the blood service facility. Thank you so much for this opportunity, and remember that not all heroes wear capes, so you can be a hero by donating blood.
Thank you very much. And now for the concluding presentation, let us welcome Mr. Christian Dale M. Soriaga to present his thesis entitled Vaccinum Myrtoides and Rubus rusticolius as Alternative Histological State.
So thank you so much for that introduction. Good afternoon, everyone, and good afternoon also to Dr. Sherwin Toriano, the Program Chair of Medical Technology, the School of Medical Technology, and also good afternoon to the technical committee involved here in the research forum who made this possible. Again, I'm Christian Dale M. Suriaga, RMT, MSMT. Without further ado, may I present to you my research paper that I had made last October 2022. The thesis title is the Vaccinia myrtoides and Rubus rossifolius as alternative histological stains. So staining is the process of applying dyes on a sections to see and study the architectural pattern and the physical characteristics of the cell. So the most popular histological stains used for the light microscopy is the hematoxylin and eosin. So hematoxylin colors the nuclei with a dark purple or blue, and eosin stains the cytoplasm of light pink cells. So plants, particularly flowers and fruits, contain this pigment called anthocyanins. So these anthocyanins provide the color of these plants. It's either a red, purple or blue. So in the Philippines, the Vaccinia myrtoides is known as ayusit, and it is a member of Ericaceae health family Cyanococcus, genus Vaccinium. It is usually found in Benguet or in the mountain province area in the Philippines. At the same time, the Rubus rossifolius or the Sampinit, it is usually found in the Quezon province. So it came from the genus Rubus from Rosaceae family. So as you can see in the picture, the red one is the Rubus rossifolius that will stain the cell as a red color or pink color, while the other one is the Vaccinia myrtoides or the blueberry. So this study was perceived to be beneficial to the medical technologies. Okay, why? That helped to perform the histological laboratory procedures on our daily basis, since they were exposed to the risk and hazard of handling commercially prepared hematoxylin and eosin. Also, medical technologists who have limited access or do not have easy access to the hematoxylin and eosin stain will be given an alternative option for histological stain. So this study was developed to found an effective and alternative for the chemically prepared hematoxylin and eosin dye. The specimen for the research were the pigment coming from the fruit of Vaccinia myrtoides and Rubus rossifolius. So the Vaccinia myrtoides will be substitute for hematoxylin stain, and the Rubus rossifolius will substitute for the eosin stain. Next slide, please. This study determined the suitability of Vaccinia myrtoides or the blueberry and Rubus rossifolius or raspberry fruit extract as alternative to histological stains. So specifically, it sought the following question. So number one, what is the staining effect of Vaccinia myrtoides and Rubus rossifolius ethanolic fruit extract when temperature interacted with time storage, when concentrations interacted with temperature storage, and time temperature interacted with concentrations. So number two, is there a significant interaction of the staining effect of Vaccinia myrtoides and Rubus rossifolius fruit extract between A, time storage and temperature storage, B, concentrations and temperature storage, C, time storage and concentrations. Next slide, please. As you can see in the slide, the conceptual framework. So the input include the independent variables. So what are the independent variables in this research? We have here the fruit extract, the different concentrations, the time storage, and temperature storage. For the process, it includes the methodology or the procedures of the research. And for the output would be the final product of the research, which is the alternative histological stain for hematoxylin and eosin. Next slide, please. For the methodology, first we have the preparation of the plant extract. So the preparation of both berries, the Vaccinia myrtoides and Rubus rossifolius, 600 grams of blueberry and raspberry were subjected to washing to remove dirt. So the other foreign particles that may affect the color of the stain. So this was placed in the juicer, okay, to separate the pure berries extract from its pulps. So the pure extract was collected and was placed so that centrifuge for 4,000 RPM for 10 minutes so that the clear supernatant was separated from the sediments and it will be stored in a beakers. Next, we have the preparation of different concentrations. So for the 25% berry solution, so 25 ml of the previously prepared supernatant from the pure extract will be added to 75 ml of ethanol. For the 50% berries, 50 ml of the previously prepared supernatant, it will be added to 50% of ethanol. Then for the 75% of berry solution, 75 ml of the previously prepared supernatant from the pure extract was added to a 25 ml of ethanol. While for the 100% concentration, the 100% berry solution, so the 100 ml of the previously prepared supernatant from the pure extract was transferred to fill a 1 coupling jar and nothing will be added to the extract. So the extract of Vaccinia myrtoides and Rubus rossifolius, when in time duration was stored, it will be stored in day 1, day 3, day 6, and day 9. While in the temperature storage, both extract will be stored at room temperature and refrigerator temperature. For the addition of the mordant, so the mordant which I used here was the aluminum potassium sulfate. So it was dissolved in a distilled water and then mixed into 1:10 ratio. So after mixing the solution, it was heated for 2 minutes and cooled down for 30 minutes. Then you will add this mordant to Vaccinia myrtoides or the blueberry supernatant. Okay. For the preparation of the histologic sample, so I use here a normal gallbladder tissue. So using the routine preparation of a biopsy sample, the gallbladder sample undergo fixation, dehydration, clearing, infiltration, embedding, cutting, and staining. So in the staining of the histological samples or staining the gallbladder tissue, the 4 tissue samples from each subject were prepared and represented for staining concentration. So including the hematoxylin and eosin, such that the first set of tissue slide was stained with 100% berry extract. The second set will be stained with 75% berries extract. The third set will be stained with 50% berries extract and 25% berries extract for the fourth set. Also with the control, which is the hematoxylin and eosin, which is the commercially prepared staining solution. So these are the pictures. Next slide, please. So these are the pictures during my experiment. Next slide, please. For the evaluation of histologic samples. So after staining, after mounting the prepared slide, the staining efficacy of Vaccinia myrtoides and Rubus rossifolius extract in a normal tissue was compared by 4 pathologies. So a criterion was followed from the study of ESIP in 2019 as cited by Ong et al. In 2017 to determine the staining ability based on the histomorphologic appearance like the nuclear staining, the cytoplasmic details, staining intensity, and contrast and clarity. The slides were then graded based on the staining intensity of the berries extract. So each of the criteria will be given a score of 0 to 4 based on the ability to appreciate the stain section. So, so this one, the table 1. So to be able to analyze the research, the researcher with the help of the statistician process the data using MS Excel and PHSAT software. So I use here the descriptive summary for the data gathering, the one-way ANOVA and the two-way ANOVA. Next slide, please. Okay, as seen in the first table, the staining effect of Vaccinia myrtoides and Rubus rossifolius ethanolic fruit extract when temperature interacted with time storage. So the main result of the temperature and time storage when interacted with the staining effect of the fruit extract considering the outcomes, it can be observed that the Vaccinia myrtoides and Rubus rossifolius fruit extract that were stored at refrigerator temperature based on a mean result, it was able to impart a good to excellent staining effect throughout the time storage. While the mean result of the fruit extract that was stored at room temperature gave a satisfactory to excellent result. So according to the research of Yusuf et al. In 2014, 4 degrees Celsius was the ideal temperature to store the anthocyanins to guarantee the pigment stability. Sorry, excuse me. For the second table, as displayed in the second table, the mean result of temperature and different concentrations when interacted with the staining effect of the fruit extract in 25% concentration that was stored in refrigerator had a mean result of 3.38, while in the 50% and the 75% concentration had a mean result of 3.44. Furthermore, the 100% concentration had a mean result of 3.69. It showed that the different concentrations that were stored at refrigerator temperature had an excellent staining effect on the gallbladder tissue. Based on the result, the different concentration that were stored in refrigerator were able to impart an excellent staining effect. In the 25, 50, and 75% concentrations that were kept at room temperature had a mean result of 3.00, 3.12, 3.13 respectively. So it indicates a good staining effect on the gallbladder tissue, while the 100% concentration stored at room temperature had an average result of 3.50, also manifested an excellent staining effect on the gallbladder tissue. So take note that temperature is another important environmental factor according to Liu et al., which affects the metabolism of these anthocyanins. Inhibiting the expression of the anthocyanin activator results in high temperature that will reduce the anthocyanin accumulation in plants, and low temperature stimulated the anthocyanin accumulation in some vegetables plant. Next slide, please. As shown in the third table above, the mean result of time storage of Vaccinia myrtoides and Rubus rossifolius when time storage interacted with the concentrations. Based on the result, the 25% concentration when stored for a longer period showed a satisfactory to good staining result, and the 50% concentration showed a good to excellent staining result, while the 75% and the 100% concentration maintained the excellent staining result even though the time was longer, where the cytoplasm was visible, clear and sharp. Cells are stained with bright, excellent staining result, okay? And intense color with the nucleus and cytoplasm. Well, the cytoplasm are well differentiated. So therefore, as the storage duration increase in the staining effect, it also intensified. So this is comparable to the ripening of the action of the hematoxylin. Why? Because where the oxidation naturally occurs as the stain and the ultimate result get aged, the substance keep its staining ultimate result for a longer time. Storage causes anthocyanin degradation, as Kopjar et al. In 2014 pointed out. Since anthocyanin are known to be prone to degradation at the lower temperature, it was hypothesized that the anthocyanin content at the ambient temperature would be lower than 4 degrees Celsius. So anthocyanin retention in extract kept the ambient temperature for 42 days, range from 61 to 81%, while it ranged from 52 to 87% if the extract kept at 4 degrees Celsius. In the fourth table, the interaction between the time temperature in relation to the staining effect of Vaccinium myrtoides and Rubus rossifolius ethanolic fruit extract was proven to be significant. At a p-value of 0.00, the null hypothesis of no interaction between the time and temperature was rejected. Going back to the first table a while ago, the combination that gave the best possible staining effect, that was the day 6 of the refrigerator temperature and day 9 of the refrigerator temperature. While in the interaction between the temperature and concentration in relation to the staining effect of Vaccinium myrtoides and Rubus rossifolius ethanolic fruit extract was proven to be not significant. So at a p-value of 0.121, the null hypothesis of no interaction between time and temperature was not rejected. Going back to the table number 2, the combination of temperature and concentration resulted to statistically no difference in staining effect of the all possible combinations. So it was concluded that the staining effect of the 25% at room temperature was statistically the same to the staining effect of the 100% concentration at the refrigerator temperature. So statistically, the same value of the 75% concentration at refrigerator temperature. So while in the interaction between time and concentration in relation to the staining effect of Vaccinium myrtoides and Rubus rossifolius ethanolic fruit extract was proven to be significant. At a p-value of 0.00, the null hypothesis of no interaction between time and concentration was rejected. So going back to the table number 3, the combination that provide the best staining effect was day 6 at 75 and 100% concentration, as well as in the day 9 at 75% and 100% concentration. Next slide, please. So this is the outcome of the staining quality of Vaccinium myrtoides and Rubus rossifolius that was stored in room temperature in different concentration. We have 25%, 50%, 75%, and 100% in different time storage also. The stored stain was stored in day 1, day 3, day 6, and day 9. So letter A represents the nucleus of the gallbladder tissue. Letter B represents the cytoplasm of the gallbladder tissue. Next slide, please. So in this slide, the outcome of the staining quality of Vaccinium myrtoides and Rubus rossifolius fruit extract that was stored in refrigerator temperature in different concentrations. Same also in the room temperature. So different concentration, we have 25, 50, 75, and 100%. And in different time storage, day 1, day 3, day 6, and day 9. Same also in the room temperature. The letter A represents the nucleus of the gallbladder tissue. Letter B represents the cytoplasm of the gallbladder tissue. Next slide, please. For the conclusion, based on the findings, these are the conclusions. So the isolated dye, fruit extract of Vaccinium myrtoides and Rubus rossifolius can be potential natural colorant of normal tissue specimens. So the extract can impart a good to excellent staining effect in the gallbladder tissue. So according to the result, the 100% concentration that was stored at refrigerator temperature was the histological stain. The histological stain reagent could be formulated in this study. So based on this conclusion, based on the result and the conclusion, the following are my recommendations during my experiment. So the stains viability can be checked for additional storage of days. To compare the effectiveness across all the tissue types, various tissue types can be utilized. So the future studies may also employ the color that was recovered in samples of bodily fluids like blood or microorganism. pH, light and oxygen, and solvents are few additional variables that can be controlled in the study, which may have an impact on the anthocyanin. Next is to try another mordant okay, can be used with the extract. So further studies on the other staining application of blueberry and raspberry ethanolic fruit extract should be done. Next slide, please. So before I end my presentation, I would like to thank the following. So to the Philippine Women's University and Dr. Sherwin Toriano for this opportunity, the staff of Hospital ng Makati Histopathology Section, Ma'am Donna, Ma'am Cecil, Ma'am Camille, for bearing with me their long hours of work for their assistance with the materials needed in my experiment. To Dr. Emmanuel Gabriel, my research advisor, for guidance as he went through the process of my research. To the members of the committee, Dr. Nini Lim, Sir Alex Bolaños, and Sir Roel Malimban for their insightful comments and encouragement. To my family and friends and my co-workers in Makati Health Department for their support. And most of all, to our Almighty Father, who is my driving force and strength in everything I did to accomplish the research. So the best color in the whole world is the one looks good on you. So you can never wrong with the color that brings out your best features and mood. So all that matter is that it's the color that brings yourself. Thank you for listening.
Thank you very much to all our presenters in sharing their research thesis. Note that for the completion of each of these topics are the challenges and difficulties that our researchers have gone through. So with that, we appreciate your effort and willingness to share your study with us to learn from. Moving forward, the awarding of Certificate of Appreciation, may we call in our program chair, Dr. Sherwin Toriano.
Thank you very much and congratulations everyone. That was indeed very helpful to our researchers. The Philippine Women's University School of Medical Technology presents this Certificate of Appreciation to Dr. Rosana Tala as our guest speaker for imparting invaluable knowledge in the Medical Laboratory Science Research Forum 2023 with the theme Research Thesis Scientific Journey from Title Through Gaps, conducted on the 26th day of January 2023. And also, the Certificate of Appreciation goes to, next slide please, Miss Irish Ponce, Mr. Christian Dale Soriaga, Miss Charmaine Grace Texon Balbin, and to our interns, Miss Rona Caril Enriquez and Miss Kimberly Marie Ibarra. Once again, congratulations everyone. You did a good job.
Again, congratulations to our speakers and thesis presenters. You have heard them all in their invaluable sharing. So if there are any questions for clarification or difficulties in your research study that our speakers and presenters may help you with, we'll take advantage of that in our open forum. So let me announce that the floor is now open for questions. May we invite once again our plenary speakers and thesis presenters to shed light on the questions presented.
MCs, do you have questions for our speakers and thesis presenters?
We do have here, sir, first for Dr. Tala. The question would be, what are the advantages of doing experimental research compared to the qualitative one?
As I've mentioned earlier, for students like us from the medical field, I suggest that we dwell on experimental research because not only challenging to us, but also we can really contribute, as mentioned by Dean SB, that we have to contribute on the improvement of health and the delivery of medical technology services to our clientele. We also can do descriptive research. However, in terms of challenges, I think we are more, the area is more applicable to areas of arts. Okay. So, para sa atin talaga ang experimentation. But both of them are challenging tasks to do. Thank you.
Thank you very much, Dr. Tala. And for the next question is for Ms. Irish Ponce. The question is, what is the function of Alaxan in your study?
So I'm sorry. So for the alloxan increases the blood sugar of the subjects. It is by disrupting the process of the pancreas by excreting the insulin and by increasing the blood sugar. That's all.
Thank you so much, po.
Okay, with regards to the question addressed to Dr. Tala, I think there are topics which are inherently can be done properly if done experimentally, rather than doing it via qualitative researches. So in our program, Master of Science in Medical Technology, we are requiring our students to conduct experimental researches. So that's why we needed dependent and independent variables for this one. As you manipulate the independent variable, it would have effects on dependent variables. Whereas on our MSHSM programs, we require qualitative research. When it comes to qualitative research, it is very exhaustive. So no research would be easier than another. Qualitative or experimental, they are of the same level when it comes to our master students. When it comes to BSMT, since we are in a pandemic, we allowed only qualitative researches temporarily. But prior to the pandemic, we require our students to conduct experimental studies as well. Now, with regards to the question directed to Ms. Ponce, I think she has to induce a higher blood sugar among mice in order to show the effect of the medicine on the chemistries on the mice. That is why you need an induction. And niacin is actually also a treatment that could be given, which has an effect on blood sugar. But other than the effect on blood sugar, it has effect on other blood chemistries as well, as provided in her presentation.
Thank you very much. For the next question is for our interns, Ms. Rana and Ms. Kim. The question is, what motivated you to do such research topic?
Yes, hello, good afternoon. For our thesis, actually, during our brainstorming process, it was at the height of the first surge worldwide of the Omicron variant of the COVID-19. And at that time, there were already decisions made by the IATF that to lessen our precautionary measures to alert level 1. So really, it was at a time that I was, as a medical technologist and a health allied path, I was really concerned with the surges that will potentially happen in our country. So I wanted to know whether we were prepared already potentially to eradicate this virus in the communities. And I wanted to know if the practices or the knowledge were enough for the community to be lenient already. But I think Rona may have other answer.
Just to add, as for our experience in our research, we ought to pursue this topic and pursue a descriptive research as a general, because during that time that we do our study, there are still strict protocols and it affects our resources, like the availability of laboratories and availability of the person that we will need for the experiment. So to be able to still conduct research, we come up with this descriptive research, and we correlate it to the problem that we experience in COVID-19 testing. Which is the reservations of some people in undergoing COVID-19 testing. Though now that we have less strict protocols, we hope that our fellow medtech students will pursue experimental researches in the future.
Thank you so much to our interns for sharing with us the difficulties that they have encountered through their research study. For our question number 4, this will be directed to Dr. Sherwin Toriano. The question would be, what do we do if we do not have enough articles for our RRLs?
Can you repeat the question, please?
The question would be, what do we do if we do not have enough article for our RRL?
Okay. So there's a little problem with your audio. So Miss Anne, so maybe you should come closer to the microphone in order for you to speak, when you speak rather. So the problem is when you encounter very low turnouts, or you don't have RRL or articles for your RRL. So you may opt to choose adjacent articles. When we say adjacent articles, they are articles that are related to our study. For example, if you are dealing with the management of face-to-face classes, there are no studies in medical technology yet, not much, if I may say, about face-to-face classes in medical technology. So you could go through the studies about face-to-face classes in nursing, in medicine, in physical therapy, in the other allied health. So that is what we mean by adjacent studies. So you could use this in your review of research literatures. Thank you.
Thank you, Dr. Sherwin. For the next question is directed to Ms. Texon. The question is, what specific area of the blood bank need quality improvement and how?
So for the specific area of blood bank that needs to be improved is for customer medyo kasi the quality that we the specific area that needs to be improved. Paulit po ulit ng question. Can you please repeat
the question is, what specific area of the blood bank need quality improvements and how?
That's for the donor recruitment, because as of now, you can see that this is the month of January. So with it is very hard for the blood service facility to get donors. That's the part of the blood service. That's the part of the blood bank that is very hard for now, because it's lean month. So there is scarcity in blood, and on improving on improving the donor recall for that is to boost we must boost the donor recall and strengthen the
promotions.
Strengthen the promotions for donating blood.
You need to have more extensive
Extensive
promotions for blood donation.
For blood donation, yes po. And that would be my answer po. The blood donor po.
Thank you so much, Miss Texon. For the next one would be directed to Mr. Suryaga. The question would be, why do we need to observe the temperature, storage, and concentrations of our solutions?
So for the storage of the anthocyanin, because in the refrigerator temperature, the anthocyanin is more stable, so it will stain all throughout if you store the anthocyanin for a longer period of time in a refrigerator temperature. While if the anthocyanin is stored it in a room temperature for a longer time, it will disrupted, or it will not color as same as in the refrigerator temperature. So as long as this anthocyanin pigment in plants, in flowers, or in fruits are stored in a refrigerator temperature for a longer period of time, it will stain. Thank you.
Thank you very much, Mr. Suryaga. And for our last question, anyone can answer this. What can you advise for the thesis researchers having a hard time on the completion of thesis?
Maybe we should ask Dr. Tala. Dr. Tala, what can you advise our researchers who are having a difficult time in completing their thesis?
Okay, just to tell you a brief story. Okay, I have enrolled my first, ibig sabihin may pangalawa, my first thesis, rather MS in PWU in the year 1995. Okay, and I got pregnant. I have difficulty of getting pregnant, but I have completed my first proposal. Okay, but I had stopped. Okay, so after a while, I tried to come back, but I got pregnant again. So I stopped again. So sabi ko, hindi to para sa akin yung MS MedTech. Then after a while, since the requirement in college is minimum is master's degree, I tried to shift to another master's degree in which I finished my MAT in science. However, the requirement where I am teaching in medical technology is really a master's in medical technology. So there's no option for me, okay, but to finish again my master's degree in medical technology. Okay. Bumalik po ako sa PWU, pero kung 5 taon lang, pwede pa akong pagbigyan ni Ma'am Nini, pero that's more than 20 years, mga anak. 20 years. So what's the option? I have to start all over again. So nagsimula po ako sa una, sa lahat ng subjects, in-enroll ko po ulit. Then, the rest is history. So alam ko, there's no turning back. There's no turning back. I have to be focused. I have to finish this, the Masters of Technology in, MS in Medical Technology. Because that's my requirement. And I got a scholarship in San Juan de Dios, in which, pag hindi ko siya natapos, I have to return everything to them. Okay? So kaya ako nag-start, okay, tinapos ko siya talaga because of that drive. Okay? Sabi ko nga, yung drive, yung inspiration, sabi ko nung dati tumalikod ako, pero ngayon hindi na ako tatalikod. I have to go and go until I have reached and graduated in the program of MS in Medical Technology. Tsaka very supportive yung aking mga panelists, Dr. Nini Lim, Dr. Gab, okay, Dr. Silverio, they are very supportive of me. Since nanoparticle that time, parang wala pang gumagawa ng nanoparticle. So, medyo mahirap.
Dr. Tala, you were one of the first that time.
Opo. So, ang hirap din po ng synthesis. Sabi ko, titigil na naman ba ako? Titigil na naman po ba ako? Pero sabi ni Doc Nini Lim, go on, kaya mo yan. So, sa totoo lang, nung pinalabas po ako, okay, nung pinalabas po nila ako after nung third
presentation for deliberation.
Deliberation, pinalabas po nila. Kasi nung pangalawa ko po, nagkaroon po ako ng problema because I have to review my protocols again. Imulit po ako ng aking experimentation. Because the pH in which dapat 3, ang ginawa ko lang ay 1. So, dapat triplicates po siya talaga. So, ano ang gagawin ko? Alangan naman po ako ng data. So, hindi po. So, I have to start again the experimentation. Can you imagine that? Okay, tawagin lahat uli yung tao, tapos mag-start uli kami from the top. Okay, kaya nung natapos po ako, lumabas kami ni Dr. Nini Lim, para po ako, iyak lang po ako ng iyak. Iyak lang po ako ng iyak that time. Sabi niyo, stop crying. Sabi ko, Doc, nako, kailangan, I have to make this one. Okay, but anyway, and the rest is history.
Yes,
at maraming salamat po sa WU.
Yes, you finished, Dr. Tala, and you are now a doctor. So congratulations for that one. And
Thank you po.
For our college students' information, when it comes to a master's degree, the standard when it comes to thesis is very much stringent. But do not be afraid, because we will help you out in completing your thesis. Now, this would be my advice to all those who would be working on their thesis in the proposal or in writing your thesis title. MSMT and HSM students, I require them to submit the thesis titles to me, at least 3. What is my reason? At least for asking them at least 3. If I reject the first one, you still have 2. If I reject the second one, you still have the third title. Now, once I have chosen one title, this one title is probably the most unique, because I search all the titles if these titles have been researched before, or research regarding these titles have been done before. Kung wala pa gaano, so that is one of my reasons for choosing that for you. Now, I read the proposal. My requirements for the proposal, for the proposed title or the conceptual paper, is that you should be including the introduction, a short introduction. Next, the statement of the problem. And the last one would be the methodology, in order for me to understand what you would want to do in your research. Now, if I would be having some questions, I would throw the questions to the proponent, to the student. How will you able to procure this material? Where are you getting this? How much is this? Do you have the resources for this? How long will it be procured? Those questions, practical questions. Another question would be, have you done a preliminary testing on this? So what is the result encouraging or not? So those are some of my questions. If you would be able to answer that, then maybe you are on your way to your proposal. However, if there would be some problems, then maybe the student is not so ready for this particular research. If you would not be able to answer some of the problems, you would find it difficult to complete the thesis. So that's why I'm asking some questions. I am looking at the feasibility of the study. I am looking at how possible can this be done. If I feel that it is difficult, it is not possible for the research to be done, then I would be asking several questions to the student. Because what is the use of accepting or approving this particular title if it cannot be processed, or if cannot be done? If it cannot be completed. Now, for those students who are trying out or writing their proposal, make sure that the methodology can possibly be done and can be completed. So if there would be research gaps in the methodology pa lang, then that would be a problem on the part of the student. So circumvent those problems before you submit a proposal, so that when I ask you, kasi makikita ko yung research gaps, I'm very particular with the research gaps. When I say the research gaps, that would be the question that I would be asking the students. If the students would be able to answer when I found out the research gap in the methodology, then good, because that would be that means that the student would be able to could possibly finish the research. But if not, then that would be another problem. That would be a problem at the end of the student, and the possibility that the student may not be able to finish the thesis. So in looking at the proposal, which includes the introduction, the statement of the problem, and of course the methodology, then you can somehow predict if the student may be able to finish or complete the thesis or not. So I rather not choose a proposal that would be very difficult to perform. I do not require proposals which are astounding. I do not require proposals or thesis titles which are only to impress. We want research proposals or titles that are contributive to the field of medical laboratory science. We want researches which would make you feel that I am a medical technologist, and this is my work and I am proud of it. So help yourself by doing something which you could probably finish. And if you encounter research gaps, try to circumvent them. If you cannot circumvent them, then that would be a problem. So you need to look for another thesis title. So I weigh the acceptability of the proposals, the thesis titles, on how they were written from the introduction up to the methodology. So if there would be some problems, I would be asking the students. And if they are competent enough to answer these questions, then they may go ahead with these particular thesis titles. Thank you very much.
Thank you so much for that wonderful and enlightening exchange. If there are still some questions, you may also approach our thesis advisors or even our very own program chair. To culminate this event, may I call on one of our esteemed graduate school professors. A warm round of applause to Dr. Billy J. Pedran.
Good afternoon, everyone. As the COVID-19 pandemic ravages different parts of the world, it has also brought unprecedented strain not only on the social and economic sector, but also on the healthcare delivery system across all walks of life. The disease has spotlighted equity problems while revealing holes in the healthcare delivery system that can have lasting effects on the patients and providers. Similarly, the academic institution was also affected. The closure of schools, colleges, and universities interrupted the teaching of students. Although school closure seems to present a logical solution to enforcing social distancing within communities, prolonged closures tend to impact the most vulnerable students. In the most academic institution, thesis and dissertation writing and its implementation were affected, from face-to-face implementation and laboratory works to an online survey and interviews. However, from a traditional and conservative research standpoint, these activities will not work and will violate research praxis. Nevertheless, the students who presented earlier proved them wrong. Regardless of what conditions or situations you are currently in, You can still do research as long as you have a heart that is yearning for change and a mind that defies the status quo. The pandemic opens many medical laboratory sciences and health systems research opportunities, as mentioned by our speakers. From clinical and experimental research on virus replication and prevention to determining the changes brought about by the pandemic in our healthcare delivery system. I'm sure our medical laboratory sciences and health systems management students built their research project foundation on those pressing issues. In developing a good research project, gaps should be identified. Dr. Toriano discussed research gaps and their vital role in knowledge and information building. Research gaps make the research novel because research gaps are unexplored and underexplored areas with scope for further research. This poses a challenge to our dear students to think outside the box and not merely replicate research, but develop novel research that will benefit the community. Gone are the days we do research for academic completion or promotion. Instead, we research because it is our duty as healthcare professionals to search for knowledge and to validate our claims. Ultimately, we do research for the benefit of humanity. As we close this momentous event, let me give you a quote from Albert Zentz Georgi. Research is seeing what everybody else has seen and thinking what nobody else has thought. Our scientific journey as a researcher does not stop here because finding answers through research is a way of life. Maraming salamat and mabuhay po tayong lahat.
Thank you, Dr. Pedron. For final announcements, kindly answer the evaluation form after the webinar. The evaluation form may be accessed through the QR code or the link flashed on the screen. The code and the link will also be posted in our official MedTech Facebook page. The certificates will be emailed to all registered and evaluated on or before February 10th.
We hope to see you in one of our upcoming virtual or on-campus events. Let us remind you to wear your mask and stay healthy in this post-pandemic era. Again, I am Aneriel Ulayao, together with my co-host Kiana Clarice Borla. It has been our pleasure to host this event for today. Thank you, everyone!